Welcome everybody. This is the Chemistry, Biology and Math Revision Hub. Today we will be looking at the Pearson Edexcel International A-Level Biology Unit 5 for October 2021. This paper is 90 marks, so I will go straight to the first question. Question 1 says, skeletal muscle is made of bundles of fibers. They say describe the role of calcium ions in producing contractions of a muscle fiber. So I will go down here. Let me use highlight. Okay. So let me go down here. There is some excerpt from your textbook that I put here to show you how the contraction of a muscle is achieved, or contraction of a muscle fiber is achieved. Now we have troponin. Troponin is formed onto the actin filament, and then we have tropomyosin. So when a contraction is to be initiated, calcium ions will bind onto troponin, as you can see here. The calcium ion is binding onto troponin, and when it binds, it causes the tropomyosin to pull away from the myosin binding sites, leaving them exposed. As you can see here, they're exposed. So this is the myosin, and that is the myosin head. When these are exposed, the myosin head is going to bind onto that side, and when it binds, then it will cause the bending forward, it bends forward to this direction. As you can see, when that occurs, the sarcomere is going to be reduced, so it's going to be decreased. Now, after that, ATP has to bind onto it so that it can cause the myosin head to return to its original position. So you are free to read through this in your free time. However, let me go to the first question that they're asking us to answer here. They say, describe the role of calcium ions in producing contractions of a muscle fiber. So one thing, like I've said already, calcium ions bind onto troponin. The troponin is formed on the actin filament. As you can see, this is the actin, that is troponin. The calcium ions are going to bind onto troponin. When they do, they change its shape. The troponin shape basically is going to change, or it's going to pull away. So when it pulls away from the tropomyosin it's attached to, this tropomyosin is also going to move away from the myosin binding site. And when it moves away, it leaves them exposed. When they're exposed, then the myosin head is going to bind onto those exposed sites, forming an actomyosin bridge or actomyosin bridges. And when this occurs, the myosin head will bend forward, moving the actin filament and shortening the sarcomere. So that is how contraction is occurring. So basically, in a contracted set, the sarcomere is going to be shortened. So I will go to the next part of the question. Part B says, the table shows some features of slow and fast twitch muscle fibers. So they've given us the features here. In the features we have number of mitochondria per fiber. We see slow twitch have more mitochondria or many mitochondria, while fast twitch have fewer mitochondria. Presence of mitochondria or many mitochondria tells us there is a lot of aerobic respiration to take place. As you guys know, aerobic respiration takes place in the mitochondria, so a lot of ATP is required from aerobic respiration. Here we also see the number of myoglobin molecules per fiber. We see for slow twitch there is a lot of myoglobin, or many, in comparison to the fast twitch. Again, this tells us a lot of oxygen has to be stored in order for aerobic respiration to be carried out. The capillary network is larger in slow twitch in comparison to the fast twitch, and that tells us that there is a lot of blood that has to be supplied containing oxygen, again for aerobic respiration. And then lastly, they say contraction. Slow twitch fibers contract slowly and they are sustained. Sustained contraction mean they enable the person basically to carry out activity for longer periods of time. However, the response is going to be slow in comparison to the fast twitch, where the response is rapid and it's going to be intense, but maybe it might not be sustainable for longer periods of time. The question they're asking here is, Olympic marathon runners have a high proportion of slow twitch fibers in their muscles. Explain how these features the features of slow twitch fibers shown in the table are of benefit to an Olympic marathon runner. So we will break down feature per feature. I said many mitochondria allows for aerobic respiration. Of course, that's it. Many myoglobin molecules allow greater oxygen storage, or allow for greater oxygen storage. And then the large capillary network ensure good blood supply containing glucose and oxygen, again for aerobic respiration. And then slow and sustained contractions allow longer periods of exercise for longer races. And again, remember, these are marathon runners. Marathon runners run longer races, so they need that sustained drive in order for them to be able to continue onto their races until when the race is done, basically. I will continue to the next part. Here they say the graph shows the rate of heat loss at rest and during exercise. So we can see the vertical axis is showing heat loss, horizontal axis is showing time, and we can see the start of the exercise. So the exercise was started at this point here, and we see when it was started, rate of heat loss increased drastically, and again it leveled off, meaning a maximum was reached. So the question is asking us to comment on the rate of heat loss during the exercise. In this kind of question, when they ask you to comment, just observe the graph or the data given and just say the overview you see. Since this is just 2 marks, you don't need to write too much. So what I saw is the rate of heat loss increases with exercise time. So basically, as time of exercise increases, the rate of heat loss increases. That's one part. And then until a maximum rate is eventually reached, that is going to be the maximum rate. And when it's reached, then the rate is going to remain almost constant. This is almost constant. It's not constant. It's almost constant because it's not a flat straight line. This brings us to the end of question 1. Let's continue to question 2. Question 2. Vitamin A is an essential component of the human diet. Rice is a staple food in parts of the world where a lot of people have vitamin A deficiency. Rice contains no beta-carotene or vitamin A. Cells in the human intestine can convert beta-carotene from the diet to vitamin A. Some soil bacteria can produce large quantities of beta-carotene. Question A says, describe how genetic engineering could be used to produce rice that contains beta-carotene. So if we want to have beta-carotene, it means we need to introduce the gene that is going to code for the production of beta-carotene as a protein that could be essentially produced in rice, basically. So what I said here, again, this is basically genetic engineering. So we need to find the specific gene we are looking for, and then we need to be able to put it into a delivery vehicle so that the delivery vehicle can be able, or vector, so that it can be able to deliver it into our intended strain of rice. So I say the gene involved in making beta-carotene can be identified. Here you could get it from any organism that has that specific gene. And once we get it, we are going to cut it out using restriction endonucleases, or some people call them restriction enzymes. So we have to make sure that the restriction enzyme we use to cut out this specific gene is the same restriction enzyme we use to cut out from the specific vector. Or if you're going to use a plasmid, then we should use the same restriction enzyme to cut both ends. So I say using restriction endonucleases, cut out the identified gene from the DNA, and then we can go and insert the gene for beta-carotene into a vector or plasmid or liposome. Now, these are all vectors. A vector can be a plasmid, or it can be a liposome, or it can be other ways in which we can use to deliver genes into specific organisms or into cells of various organisms. So after inserting it, so here we will insert the plasmid, the vector basically, whether you use the plasmid or liposome, into the rice cells. Remember, the vector is acting as a delivery vehicle, sending the genes or the DNA we have cut out into the cell we intend to change. So in this case, it's going to be used to basically deliver the genes or DNA we have chosen into the rice cells. Here we can use heat shock or micro-injection or a gene gun basically as well. And then next we can grow the rice that has been genetically modified, and use suitable selective method or selection method to select for the modified rice. Now, after this experiment of introducing the new genes into the rice, we are not sure if all the rice has been transformed. So basically, we have to find a suitable selection method so that we can confirm that 100% that specific strain of rice has been transformed. So a suitable selection method should be selected so that we can be able to find out if the rice has been modified, and so that could be good. Modified plant rice that have a high yield of beta-carotene. So basically, the modified plant should be able to produce a high yield of beta-carotene in order for us to get what we desire to get. This is all about transformation. Get a specific gene, find a specific delivery method, insert it in, and then find a way in which you are going to deliver that into the intended cell. And once it's in there, find a way of confirming if the cell has actually been transformed or not. If it has been transformed, then we have to look at the yield, productivity, how much of the, in this case, protein is being produced. If we have two strains of rice, the rice that is going to have the highest yield of beta carotene, that could be the rice we go with, because it's going to produce a much more beta carotene, which is vital for production of vitamin A. So part B says, describe the possible risk of growing genetically modified rice. Now, when we grow genetically modified rice, of course, the biggest risk is not knowing the long-term side effects. So, of course, this genetic engineering has not been here for a longer period of time, and until now, we are not sure of the long-term side effects of specific things. So, here are long-term side effects to the environment. We are not sure if these specific genes could leak out and be incorporated into other species found in the environment, and so that could be a problem. Or it could be the environment, or it could be to other species due to the rice being genetically modified. Another thing is accumulation of genetically engineered material, non-targeted. This is, of course, accidental. What if we are modifying this rice? However, of course, genes could leak out, non-targeted in the environment. That could also occur. There is crossbreeding that can occur between the different strains of rice, or between even rice that are closely related. When crossbreeding can occur with other rice species, or with pollen transfer within the same species, that could lead to production of a strain we were not prepared for, and that could be problematic. So that is a risk. Lastly, it can lead to production of superpests or weeds. If you have genetically engineered, here they're asking about genetic engineering, not only to introduce beta carotene, but genetic engineering of rice. What if we are trying to genetically engineer rice that is resistant to pests? It means some of the genes that are introduced to this rice could potentially leak out into and be received by some weeds. And once those weeds have that ability to resist pests, it means they can also grow and compete with the photo plants we are interested in growing, and that would be problematic for the farmers, basically. So the next part is to suggest how one of these risks could be reduced. The risk I chose was basically to reduce production of superweeds. Reduction of superweeds. If you grow the genetically modified and non-genetically modified rice separately, that will ensure that they are not combining, or there is no gene flow from one type of rice to another type of rice, and that can minimize the risk of unintended or non-targeted transfer of genetic material from one specific type of rice to another type of rice. So grow them separately. The modified and the non-modified should never be mixed in order to prevent mixing up of genetic material from the environment. This brings us to the end of question 2. I will go on to question 3. Question 3. The mammalian retina is a tissue that detects light. Rod cells and bipolar neurons are cell types found in the retina. What is the name of the chemical formed when light is absorbed by the photopigment found in rod cells? Now, the photopigment found in red cells, of course, we have rhodopsin. So when rhodopsin absorbs light, it's going to be converted into opsin and transretinal. So, of course, in rhodopsin, we have opsin and cisretinal. It's going to be converted into opsin, which was already there, and the transretinal. So the cis is going to be converted into trans. That is why the answer here should be transretinal. And then the next part says, which row in the table shows the effect of light absorption on cells in the retina? So rod cells, when they absorb light, they will actually be hyperpolarized. Yet the bipolar neuron is going to be depolarized because, of course, so this is going to be hyperpolarized because the sodium ions are going to be prevented from entering inside the cell. Yet here the sodium ions are going to be entering the cell. So here we're going to have hyperpolarization, while here we're going to have a depolarization. So the answer here should be a B. I will go to the next page. They say an investigation studied the effect of light intensity on the mammalian retina. Samples of retina were exposed to different light intensities, and the mean potential difference of the bipolar neurons was recorded. The results are shown in the table. So here we have the light intensity, and then we have the mean potential difference. Okay, so the first question says, describe the effect of light intensity on the mean potential difference of bipolar neuron. As you can see, moving from the up here to down, we see the light intensity increases, and we see there is an increase in mean potential difference until this point here, until here. So we can say The mean potential difference increases with increase in light intensity up to 12 AU. You can see until here. Then the mean potential difference remains at 20 millivolts, which is going to be constant after the light intensity reaches that. So basically after that, it will remain constant no matter the increase in light intensity. That is how we can describe what is occurring here. The next part says, calculate the percentage change in mean potential difference when the light intensity increases from 4 to 12 AU. I will go back here. The change from 4 to 12 causes a change from 17 to 20. So the change is going to be 20 minus 17, which gives us a 3. So mean potential difference is going to be 3. So the percentage change will be 3 divided by the original, which was 17 times 100, giving us a 17.65%. That is the percentage. I will go to the next part. The next part says, give 2 reasons why some people might have objections to the use of mammalian retinas in this investigation. Now, if you use mammalian retinas in the investigation, it means you have to cut out tissues from mammals, basically. And this, when somebody's retinal tissue is cut out, there is a chance that it can lead to blindness. So that is very bad if they are still alive, basically. Also, it's unethical because usually when you carry out research in biology, you have to weigh the benefits as well as the harm. The pros and cons, you have to weigh them. So if the benefits are better than the shortcomings or the problems the experiment causes, then we could go with it. But in this case, the benefits do not outweigh the harm caused to the animals, so this will not be unethical. And then lastly, of course, sometimes when they are cutting out mammalian retinas, they have to use anesthetics. When they do these anesthetics in surgical preparation process, there is a chance that there is some harm that is going to be, so the animals will experience some harm, or some harm is going to be caused onto these animals, which is not a good idea. Lastly, which statement describes the response of the muscle in the iris to increasing light intensity? Of course, iris have the circular muscles as well as the radial muscles. So when there is increase in light intensity, the circular muscles are going to contract. They do contract so that the pupil can decrease, and the radial muscles are going to relax. So the contraction of the circular muscles and the relaxation of the radial muscles causes the pupil to constrict, which limits the amount of light that enters the eye. So the answer here should be C. This brings us to the end of question 3. Let's move on to question 4. Question 4, homeostasis is the state maintained by living systems. So they say feedback mechanisms are involved in maintaining homeostasis. Describe the difference between a negative feedback mechanism and a positive feedback mechanism. Negative feedback mechanisms actually work to reduce the change in a specific variable. I'll give an example. If temperature increases, the negative feedback mechanism works to reduce the temperature, and if the temperature increases, it works to increase the temperature. However, when I say reduces the change, it means if there is a change, it minimizes that change or the change in a specific variable so that it can return everything to equilibrium. Now, for positive feedback, on the other hand, it works by intensifying the change in a specific variable. Let's see some examples in positive feedback. We can have blood clotting. Blood clotting. When the blood begins to clot in the body, positive feedback works to intensify the clotting process. Another thing could be during childbirth. I'll say labor. During labor in childbirth, when this occurs, of course, the contractions are going to be intensified. Contractions are going to be intensified. And that means, of course, that is going to be positive feedback. So those are the 2 examples of positive feedback. And the negative feedback, they are all the other feedback mechanisms you study in your book, controlling blood sugar levels, controlling water levels in the blood. All those are, of course, negative feedback mechanisms. So let's continue to part B. They say, which statement about homeostasis of thermoregulation when the human body temperature is 37 degrees Celsius is correct? So they say, A, thermoregulation is no longer required. No, thermoregulation will always be required no matter the temperature, because any slight change could be catastrophic. The body no longer generates any heat. No, the body will be generating more heat than it can lose. No, the body temperature is maintained in dynamic equilibrium. Yes, this is very important. Dynamic equilibrium ensures that there is going to be a state of constant back and forth. That is dynamic, okay? So the answer should be D here. Next we see they say the health of a kidney can be determined by measuring the glomerular filtrate rate, GFR. This method measures the rate at which a small molecule called creatinine is filtered from the blood basically. They say explain how small molecules like creatinine are filtered from the blood in the glomerular. And again, they said small molecules, so we have to focus on the size and how they go through. So I said, since here we see that creatine is its molecule basically is a small size, it can be forced through the pores of the basement membrane using the high pressure in the glomerulus. This is a 3-mark question, and what they're looking for is you acknowledging that it's small in size and it passes through the basement membrane, and pressure is going to be created in the glomerulus that causes it to pass through. So that is how you get the 3 marks there. The next part says, in 15 hours, a person excreted 160 milligrams of creatinine in the urine. Calculate the GFR for this person, and then give your answer to spread significant figures. Remember the time is 15 hours, and then the amount is that. So they want us to get the value. So of course I'm going to get it. They wanted us to get the value in a milligram per minute. So the 1960 milligrams divided by 15 hours is 15 times 60. So this is converting time into minutes. When you multiply 15 times 60, you convert it to minutes. So when you divide, I get 2.18 milligrams per minute. And since they say 3 significant figures, the answer has to be 2.18 because in 3 significant figures. I will go to the next part. They say in type 2 diabetes, body cells take up less glucose in response to insulin. Many people with type 2 diabetes are overweight or obese. Some of these individuals have been given surgery to help weight loss. A clinical trial investigated the effect of surgery on the uptake of glucose by body cells in the presence of insulin. This trial used groups of people who were non-diabetic, people in the early stage of diabetes, meaning pre-diabetic, and people who were type 2 diabetic. So let's continue. Here we have the results in the table. Now the key thing here is they've given us the results, and since there is the standard deviation, it means the experiments were repeated and the means were calculated, so the results can be dependable. For each, you can see we have the standard deviation that shows us experiments were repeated. The standard deviation was calculated. Results could be more reliable. So we can go. The first part they say, comment on the results of this trial. So let's see. Overall, we can see that surgery actually improved the rate of glucose uptake among all groups, because you can see before surgery, after surgery, there is an increase for all. Again, this increase I calculated here. It's not including the standard deviation. It's just that value and that value. I use the mean, changing the mean. So this mean to that mean to that mean, and then this mean to that mean. The difference is what we can see. So because there is an increase, a positive increase in all the experiments that were carried out, we can say that there was an increase in uptake in all groups. And then here we can see that for non-diabetic, or those who are not diabetic, there was an increase. You can see the increase was basically 0.32, and for pre-diabetic, we can see a 0.75, and for type 2 diabetes, which is 0.70. Acknowledging these mathematical figures can earn you marks, so that is for marking point 2. And then the third marking point, I can say that there is an increase in standard deviation for all. You can see even the standard deviation increased for all groups, making the data before surgery more reliable. If you look at the error bars, let me use an example. So this result here was basically 1.07. The highest value was 1.07 plus 0.29, and the lowest value was 1.07 minus 0.29. That is giving you the range of values. However, when you look at this data here, it's going to be 1.39. Let me erase. 1.39 is the mean. The highest value is 1.39 plus 0.58. If you can look at this figure and that figure, it means there is a change. The standard deviation increase for all. The change from the highest and the lowest value, that difference between the highest and the lowest value is really great, or it became greater after surgery, meaning the results are not that reliable in comparison to the results before the surgery. And lastly, I say the greatest change after surgery was among the pre-diabetic group. Of course, you can see those are the ones who have the change of 0.75 in their results of glucose uptake. So I'll continue to the next part. Calculate the difference that surgery makes to the mean uptake of glucose in the non-diabetic group and the type 2 diabetic group. So we can see from the previous page you saw that the difference for the non-diabetic group was 0.31 and for the, of course, type 2 diabetic group is 0.70. So if we are looking for the difference, the surgery created between the non-diabetic and the type 2 diabetic group, of course, it's going to be 0.70 minus 0.32, and the difference is going to be 0.38, which gives us the answer here. So this brings us to the end of question 4. Let's move on to question 5. Question 5. Animals use aerobic and anaerobic respiration to produce ATP. Aerobic respiration has 3 stages where reactions occur between enzymes and substrates. So here they say which row in the table shows the location of each stage. So the 3 stages are glycolysis, the link reactions, and the Krebs cycle. So of course, glycolysis occurs in the cytoplasm. The link reactions do not occur in the cytoplasm, they occur in the matrix of the mitochondria. And Krebs cycle also occurs in the mitochondria matrix. So the answer is B because it basically all the 3 parts are marked, so it's B. The next part says a student calculated the volume of oxygen used by a cockroach as 1.7 centimeters cubed per hour. The same cockroach produced 1.3 centimeters cubed per hour of carbon dioxide in the same time. What is the respiratory quotient of this cockroach? Respiratory quotient RQ should be the volume of CO2 out divided by the volume of oxygen in. So here we can see since we have 1.3, that is going to be produced divided by 1.7, which was taken out, and it gives us 0.76. Now when we look at these answers, 0.7 is not 0.76. You round off to the closest, so 0.76 is approximately 0.8, and therefore B is going to be the answer here. So I will continue to the next page. They say hydramethylnone is an insecticide that is used to treat termite infestations. So it inhibits the electron transport chain, as you can see here. Which row of the table shows the expected changes caused by the hydramethylnone? So the first part, they say oxygen consumption and lactate production. So because it's inhibiting the electron transport chain, the oxygen consumption is going to decrease since our reactions from the electron transport chain lead to the use of oxygen where oxidative phosphorylation is going to occur. So it means oxygen consumption is going to decrease, and that means lactate production is going to increase, because if oxygen consumption decreases yet the body needs energy, it means anaerobic respiration is going to take place. And when anaerobic respiration takes place, of course, lactate production is going to increase. So here the answer should be B. The next part says, the table shows the oxygen consumption and body mass of 2 animals. So we have the animals are the southern elephant seal as well as the field mouse. The oxygen consumption is, you can see here, 1.82 times 10 power 4, and here 20, of course, centimeters cubed per hour. The body mass, of course, the elephant is really, really huge in comparison to the mouse. So they say these figures can be used to calculate the metabolic rate of each animal in centimeters cubed per gram per hour. So let's continue. They say calculate how many times greater the metabolic rate of field mouse is compared with that of the southern elephant. So we have to calculate the metabolic rate of the mouse. Metabolic rate of the mouse is going to be the oxygen, of course, consumed divided by the body mass, which gives us 2 centimeters cubed per gram per hour. And again, we are trying to find in volume per gram per hour. Those are the units we want to use, so that's why I divided the 20 centimeters cubed per hour per gram to create the units we're looking for. Then the metabolic rate of the elephant is going to be 1.82 times 10 power 4 divided by the body mass, which is 5 times 10 power 6, and it gives us that. If we compare the two, you can see that actually the mouse has a higher metabolic rate in comparison to the elephant or the elephant seal. So how many times higher is that metabolic rate? So divide the metabolic rate of this, divide it by the metabolic rate of that, and we get 549.45. Usually, it's better you give the answer to significant figures, referring to those that were already given. If you see here, we can see 1.82, 5.00, so we can try to stick to that 5.49 times. So giving a whole number is going to be better in this case, and since we cannot round off the 4, just say 5.49 times. However, they could accept other rounded off versions like 549.5. They could accept that as well. So I will go to the next part. The next part says, the photograph shows a crocodile that lives in saltwater habitats. So this is the crocodile you're looking at. They say an investigation measured the lactate production rate of these crocodiles when they were active. The graph shows the results of this investigation. So as we can see, there is lactate production on the y-axis and body mass on the x-axis. And since the data is spread all around, but they've drawn for us a specific curve to show to us that overall lactate production is it increased a little and then tried to decrease as body mass increased. So overall, there is a trend, a downward trend like that. That is what we can see from the graph. So let's go to the next page. Each plotted point on the graph represents a single crocodile. Discuss the role of the different types of respiration in the active crocodiles. So when they say the different types of respiration, they mean aerobic and anaerobic. So we have to discuss the role of aerobic and the role of anaerobic. And then next they say, use the information in the graph. So we will refer to the graph and then your own knowledge to support your answer. So we are going to refer to the graph and we are going to refer to the theory, of course, the things you have already read, talking about the types of respiration in detail. So I answer this question as follows, again referring to the graph that we have. Before I answer, let me first take you back. So here we have this graph we were given. We can see that overall lactate production actually decreases as body mass increases. We can see this overall. However, we can see there is no direct relationship. I cannot say this is a positive correlation or it's negative. I cannot say there is no direct relationship between the body mass and lactate production, because you can see some of the smaller crocodiles produce the same amount of lactate as the bigger or larger crocodiles. So there is no direct relationship between body mass and the lactate production. That is the second point we can see here. And the third point we can see is this one here. This is about 20 kilograms. The body mass, 20 kilogram body mass crocodile produced the highest rate of lactate. So that is another observation we can see here. And since there is lactate production, it tells us that there is anaerobic respiration going on. Since anaerobic respiration is going on, we can know that there is intensity in exercise, meaning the crocodiles are exercising intensely, and when they do exercise intensely, there is more lactate that is going to be produced due to anaerobic respiration, and this will occur because the aerobic respiration pathway will not be adequate to produce enough ATP required to sustain these the exercise activities that crocodiles are going through. So I will go back to answer the question. So the first answer I wrote, I said the larger the size of the crocodiles, the lower the rate of lactate production. This is a fact from the graph. And then the next part, the 20 kilogram crocodile had the highest rate of lactate production. Again, I showed you this from the graph. And then I also showed you this. There is no direct relationship between the body mass and lactate production, because we observe that some of the smallest crocodiles have the same lactate production as the larger ones. This point is backing up that point, backing up the data from the graph. And then as we go on, as the crocodiles exercise vigorously, or as the intensity of the exercise increases, aerobic respiration or aerobic pathway will not be enough to keep up. It will not be generating enough ATP, so ATP will begin to be generated anaerobically in glycolysis. And when this occurs, the stored glycogen is going to be broken down into the muscles to produce glucose, which can be used in the production of ATP. Now, since the question asked you to use your own knowledge here, use your own knowledge, now we can bring in our own knowledge. Our own knowledge here we know is, if glycogen is broken down to produce glucose, 1 mole of glucose is going to be converted into 2 moles of pyruvate, and then 2 net ATPs and reduced NADs. These are going to be produced. So during vigorous exercise, because anaerobic respiration is occurring, the products from glycolysis are going to be used in anaerobic respiration. And in this case, the pyruvate is going to be converted into lactate, And this process uses up reduced NAD, producing the oxidized NAD. This oxidized NAD is later used to convert lactate back into pyruvate when oxygen is actually supplied. So here we can see this is our detailed explanation. This one could earn you about 2 marks, your own knowledge on explanation. And this brings us to the end of question 5. Let's continue to question 6. Question 6. When activated, T lymphocytes can divide rapidly and secrete proteins called cytokines. So they say these cytokines will activate different types of lymphocytes. Question A1 says, which is the function of tRNA in cytokine synthesis? So you guys know tRNA, of course, it's going to bring the specific amino acid to the ribosome in order for the protein or for the polypeptide chain to be made. So it identifies the amino acid and transports it to the ribosome. That should be the answer. Then the next part says, which organelle is involved in chemically modifying cytokine proteins before they are secreted? Modification of proteins is carried out in the Golgi apparatus before they are secreted, so it should be the same. Vesicles do not modify. Smooth endoplasmic reticulum is usually into lipids. The ribosomes, no, these are for making proteins, so it should be the Golgi apparatus. Part B says, describe the role of DNA polymerase in DNA replication during cell proliferation. Cell proliferation means rapid increase in the number of cells or rapid cell division. DNA replication is basically increasing the number of DNA, and DNA polymerase is the enzyme that causes the DNA replication. So I say DNA polymerase adds complementary nucleotides. Complementary nucleotide, it means there is already a strand and it's adding nucleotides that are complementary to the nucleotides on the other strand. So that strand is going to be the template strand. So leading to the formation of phosphodiester bonds. Phosphodiester bonds are formed between different nucleotides that are on the same strand of DNA. So it also helps in perforating the DNA. DNA polymerase attaches the nucleotides that are complementary to the nucleotides found on the template strand. It also carries out perforating or editing of DNA, searching for errors that have been made in order for them to be corrected. So let's go to the next question. It says, explain how cytokine gene can be expressed in an activated T lymphocyte. Expression of a gene, of course, it will occur due to when transcription occurs. So I said, activation of a gene results in production of transcription factors. Transcription factors will bind to a specific region, we call it the promoter region, on the cytokines gene. And again, they're asking how cytokine genes can be expressed. This is about gene expression. If we have a specific gene, how is it going to be expressed? When there is a transcription factor that could bind, when the transcription factor binds, it kind of directs the RNA polymerase to come and bind onto the specific region. The transcription factor binds to the promoter region, and when it does, RNA polymerase knows that it's time for me to bind, so that transcription of the specific gene can occur. And when transcription occurs, messenger RNA is going to be produced, and this later leads to the production of the cytokines, which will be the protein. So, transcription factors, transcription occurs, and then the protein is going to be produced. That is how this should be answered. Part D says, T lymphocytes produce different types of cytokines. Some of these cytokines activate T killer cells, some activate B cells. Explain why different cytokines can activate different cells. Now, you remember cytokines have to have a specific shape, and because they are proteins, so I say since cytokines are proteins, they have a specific shape. And having a specific shape, since they're activating cells, it means there is a receptor found on that specific cell or the membrane of that specific cell that this cytokine is going to activate. So it means different cells are going to have different receptors. So it means a specific cytokine cannot activate every cell because there are different receptors on every cell. So we need to have cytokines that have complementary receptors, or whose binding sites are complementary to the receptors found on the cell membranes of the cells these cytokines have to activate. So I said they have specific shapes, and the cytokines bind onto the receptors found on the cell membranes of the different lymphocytes. So then the cytokines initiate transcription and translation of the protein in either B cells or T cells. Remember, it's after cytokines bind that these cells are activated to begin performing their function. So it means there is a receptor, and this cytokine has a shape that is complementary to that receptor. When it binds, it causes reactions to occur inside that cell. These reactions could be transcription. So basically, the binding of this cytokine could be acting as a transcription initiator, causing transcription as well as translation to occur inside the cell. And since we have T cells and we have B cells, they have different receptors on their surfaces, and therefore they have to be activated by differently shaped cytokines in order for them to be able to perform their function. And that's it basically. I will continue to the next part. So they say CD45 is a protein found in the surface of lymphocytes. Different forms of the CD45 protein are found on the surface of different lymphocytes. Describe how different forms of CD45 protein can be produced from a single gene. Different versions from a single gene. So here it means we have to carry out splicing. So because of course you're using the same gene and making different proteins out of that gene. If you remember from your AS biology, you talked about how one specific gene was used to actually produce so many different for the chickens. I think there is an example of a chicken where the different chickens hear different ranges of sound, and that is because of the proteins found within their ears. While these proteins are all made from one single gene, how were they able to produce all the different proteins from one single gene? It's because of spliceosomes. So here we can say that gene coding for CD45 gene is transcribed into pre-messenger RNA. The pre-messenger RNA is then going to be modified, so the spliceosomes remove the introns from the pre-messenger RNA, leaving exons. Then these exons are going to be rearranged to produce different messenger RNA molecules. These produced messenger RNA molecules are the ones we call the mature messenger RNA, and then these different messenger RNA molecules are then translated into different proteins. So this is called post-transcription modification, where the produced messenger RNA out of transcription is going to be modified by cutting out the introns, leaving the exons, and then the left exons reassembled or rearranged in different versions to create a specific mature messenger RNA that will leave the nucleus and go to the ribosome in order to produce various proteins, basically. So the rearrangement of these exons, it can vary, and therefore we can produce various proteins from just one specific gene. This brings us to the end of question 6. I will go on to question 7. Question 7. Researchers investigated how groups of rats treated with drugs responded to stress. Groups of rats were stressed by moving them into a new environment. The rats were then treated with a salt solution, drug A or drug B. So here we can see the table showing us the experiments that were done. And of course, we expect sending them to a different environment is going to stress them by causing an increase in their heart rate. So they say the graph shows change in mean heart rate measured over a 15-minute period. So the vertical axis shows us the change in heart rate in beats per minute, and the horizontal axis shows us the time, of course, in minutes. So the first question says, calculate the percentage difference in the change in heart rate when using drug B between 5 to 12 minutes. So this is drug B. Here we can see at 5 minutes we see we have 30 beats per minute, and at 12.5 minutes we can see we have 57.5 beats per minute. So when they ask us to calculate the percentage difference, we have to get the difference first, which is 57.5 minus 30, giving us 27.5, and the percentage difference will be that value divided by the original value, 30 times 100, giving us a 91.67%. So I will go to the next page. The next page says, question B, compare and contrast the effects of drug A and drug B on the heart rate during the investigation. So we have to give the similarities as well as the differences that are occurring. The similarities we can see is that both drugs affect the heart rate over time. At least there is a change that occurs to the heart rate when both drugs are administered. And then we can also see that the greatest change in heart rate is going to occur initially in the first 2 to 3 minutes after administering the drug. That is the similarities. The differences we can see that drug B reduces the heart rate more than drug A, and then the change in the heart rate with drug A levels off. If I can take you back slowly, you can see with drug A, it levels off at about this time. It's kind of leveling off, while that of drug B is continuously increasing. There is still an increase. It could go on and on, but since the experiment was only carried out for 15 minutes, we cannot see a point where the graph leveled off, and therefore we can conclude that this levels off, but that did not yet level off. It's still increasing even after 15 minutes. So let me take you back. Okay, so the change in heart rate with drug A levels off between 7 to 10 minutes, while that in drug B is still increasing after 15 minutes. The last part of question 7 says, explain the effect of these drugs on the control of the heart rate. So since we know that the stressful situation that the mice or the rats experienced caused them to basically their heart rates to increase, so stress increases the heart rate, and when this occurs, the brain is going to send impulses from the cardiovascular center to the sinoatrial node in the heart, and this is going to cause the heart rate to increase or to go up. How does this occur? The sinoatrial node is going to send electrical signals to the atria, and these signals will further spread out into the ventricles, causing them to contract faster, and therefore the heart rate will go high. However, when the drugs are used, the change in heart rate is going to be decreased because these drugs work by causing a decrease in the stimulation of the impulses or the electric signal coming from the SAN to the atria as well as the ventricles. This will inhibit or prevent the increase in heart rate, and therefore it will kind of bring down the heart rate or cause a change in the heart rate. And finally, we can see drug B has the greatest inhibiting effect on the sinoatrial node. So if there was further experiment to be carried out, drug B would be a good candidate to be used. This brings us to the end of question 7. I will move on to question 8. Question 8 is about the scientific articles. So I will begin by showing you the scientific articles. So let's begin with the article. I am going to slowly move. I'm not going to read the whole article because it's really, really big, but I'm going to peruse through slowly so that you guys can have a glimpse to read what is occurring. I'm showing you through all the paragraphs that are written. So this is the first page. I will go to the next page, showing you from paragraph 6. So let's go down there. We can see until 16, and then moving on to the next page, we can see 17 through 23, and then slowly we go to 26, and then the next page. So this is the last part of the scientific article. So we're going to use this to answer the questions that are on the next page. So let me go to question 8. Question 8 says, the scientific article you have studied is adapted from Rethinking Alzheimer's in New Scientist. So it was published in February 2019. They say use the information from the scientific article and your own knowledge to answer the following questions. So the questions are, memory and cognitive function are two processes that occur in the brain. This is paragraph 2 and paragraph 13. I have transferred some of the paragraphs into the pages of the questions so that we can read them as we try to answer the questions. So they say, different parts of the brain are responsible for specific functions. Complete the table showing which parts of the brain are responsible for the different functions. So this is kind of easy. The cerebellum, this one here is for coordination of voluntary movement, balance, as well as maintaining posture. So any of these would be okay. Now, if they ask about something to do with interpretation of information from the retina, that is going to be the cerebral hemispheres, or you could say the occipital lobe. And then if they ask about the medulla oblongata, this is where we find the cardiovascular center, the respiratory center. That is basically concerned with respiration as well as heart rate, breathing in and out. Secretion of ADH, this is the posterior pituitary gland, or you would say, sometimes they accept if you say a hypothalamus, but it's the posterior pituitary gland. And again, you remember the kind of hormones that are produced in the pituitary gland. We have the anterior, we have the posterior. You need to remember all those hormones that are produced from the different areas of the pituitary gland. So here I wanted to put an image for you to show you where the occipital lobe is. So you can see it's here, down here, where we have the visual cortex. So I will move on to the next question. The next question says, some forms of dementia can be diagnosed using brain scans. Compare and contrast the use of functional magnetic resonance imaging, fMRI, and computed tomography scans, which is CT scans, to identify structures like plaques in the brain. Paragraph 6. So I'm going to take you to paragraph 6. I brought it here. So it says, some have begun to question the amyloid hypothesis. The lack of results has been compounded by the discovery that people, including some in their 90s with exceptional memories, can have brain plaques and tangles without having dementia. In a review of the research to date last year, Bryce Visor at the University of Technology, Sydney, Australia, concluded that there isn't sufficient data to suggest that amyloid has a central or unique role in Alzheimer's. So the question is asking us to compare and contrast. Okay. So, of course, compare and contrast, we have to give the similarities as well as the differences. So, similarities between CT scans and fMRI, both are neuroimaging techniques with high resolution. So, basically, their resolution is really great. And both images the areas of the brain with plaque. So, basically, the image areas of the brain that have plaques. So, both of them can do that function. The only difference is for fMRI, it uses magnetic resonance, while CT scans use X-rays. Another thing is fMRI will show oxygen uptake during brain activity. Remember, this one is functional. You can observe brain activity while the person is and the person's brain is being scanned. So fMRI will show oxygen uptake during brain activity, while CT scans only show the brain structure. Another part, you could say they are expensive, the others are not, or in comparison to the others. But these two major differences are key in you getting the 4 marks in this question. So let's continue to the next part. Part C suggests how the amyloid protein may prevent bacterial infection in the brain. Paragraph 7 and 22. So paragraph 7 said, in 2016, researchers discovered that amyloid seems to function as a sticky defense against bacteria. Now, this is very important, sticky defense against bacteria. They found that the protein can act as an antimicrobial compound that kills bacteria. And when they injected bacteria into the brain of mice engineered to make Alzheimer's protein plaques developed around the bacterial cells overnight. So meaning if the plaques develop, it means they're causing maybe the bacteria to stick onto each other as a mechanism of destroying the bacteria. So 22 says, as to how P. gingivalis might cause dementia after it arrives in the brain, there are 2 clear possibilities. It may trigger the release of amyloid, the brain's method of trying to contain the infection, and this may then kill neurons. So we can see from these 2 paragraphs, in relation to the answer, remember they're asking us how amyloid proteins may prevent bacterial infection in the brain. From these 2 paragraphs, you can see that they can cause bacteria to stick together, and then when they stick together, phagocytosis could be enhanced because many bacteria being stuck together, phagocytosis can occur consuming many in one take. And then since it acts as an antibiotic, remember they have said antimicrobial, so antibiotic, it is going to be able to destroy them. And another part, the amyloid protein could disrupt the ability of bacteria to adhere directly onto host cells. So that if it cannot adhere directly onto the host cells, then it cannot infect them, and there is a prevention for the bacteria to enter. So prevent bacterial entry into the cells. So here we are looking for 4 marks. So it could prevent bacteria from entering cells. It could it could disrupt the ability of the bacteria to adhere onto the host cells. It could be antibiotic, meaning producing some chemicals that could be or that could destroy the bacteria, and causing bacteria to stick together could further enhance phagocytosis, consuming vast numbers of bacteria within a small period of time. So the next question says, explain how the bacterium Porphyromonas gingivalis that causes gum disease may invade the brain and cause inflammation in brain regions affected by Alzheimer's disease. So here they are referring us to paragraph 10, 20, and 21. I try to put them here. They're really big, but I try to squeeze them in here so that we don't have to go back to the scientific article every time we want to try to read. So part 10 says, or paragraph 10 says, multiple teams have been researching Porphyromonas gingivalis, the main bacterium involved in gum disease, which is a known risk factor for Alzheimer's. So 4 teams have found that P. gingivalis invades and inflames brain regions affected by Alzheimer's. The gum infections can worsen symptoms in mice genetically engineered to have Alzheimer's, and that it can cause Alzheimer-like brain inflammation, neurodamage, amyloid plaques in healthy mice. Then 21 says, the blood-brain barrier should protect your brain from microbes, but P. gingivalis can evade white blood cells and other cells lining blood vessels, so might cross it that way. It may also invade cranial nerves near the mouth. This is the key part. Near the mouth, then spread from cell to cell toward the brain over a period of years. So if they can spread from the mouth to the brain, across the blood-brain barrier, it means they can be able to enter into your brain and cause some infection. Then 20 says, this inflammation can lead to chronic periodontitis and tooth loss, and some studies have shown that people with fewer teeth are more likely to have dementia. The inflammation and toxins caused by P. gingivalis damage the lining of your mouth, which may make it possible for oral bacteria to enter bloodstream and then other organs. Even if you don't have disease, transient damage to your mouth lining from eating or toothbrushing can let mouth bacteria into blood, says Lynch. So actually this comes after that, before that. This comes before that. So we can refer to this and try to see if we can answer this question. The question said, explain how bacterium Porphyromonas gingivalis that causes gum disease may invade the brain and cause inflammation in brain regions affected by Alzheimer's disease. So the key thing here is this bacteria is going to be carried in the blood from the infected gums. Remember, it could come from your mouth. That is from these paragraphs. So it's going to be carried to the blood, and then it will penetrate the blood-brain barrier. When it does, so the products from the bacterial cells, that the chemicals or the molecules that are going to be released by the bacteria can cause the release of histamines in your brain. And this can lead to vasodilation, the swelling, and white blood cells are going to increase in size in the brain, and this could lead to basically the destruction of some of those regions. And again, this question is asking how bacterium caused gum disease may invade the brain and cause inflammation. In relation to inflammation, when the bacteria leave your gums to your blood, into the blood-brain barrier, their presence causes some chemicals to be produced. Those chemicals cause the production of histamines. When histamines are caused or are produced, basically, they cause the increase in size of that blood vessels so that more blood is sent to those areas where the infection has occurred. Part E says, suggest how gingipains may have a degrading effect on memory of a person suffering from Alzheimer's disease. We have to look at paragraph 12. So I extracted paragraph 12. It's here, so I'll read through. They said, now researchers from Cotexme several universities have reported finding that two toxic enzymes that P. gingivalis uses to feed on human tissue. In 99 and 96% of 54 human Alzheimer brain samples taken from the hippocampus, a brain area important for memory. These protein-degrading enzymes are called gingipains, and they were found in higher levels in brain tissue that also had more tau fragments, and that's more cognitive decline. So if there is more cognitive decline, it means these enzymes are actually participating in the breakdown of proteins as well as tissues. So they say, suggest how the gingipains may have degrading effects on the memory of a person suffering from Alzheimer's disease. So here I say gingipains are enzymes. Of course, they've told us they're enzymes that break down proteins in membranes. So they degrade the membranes, and this will lead to loss in cells. So cells are going to be lost, and then brain tissue is going to be lost. Now remember, a group of cells creates tissues. If cells are lost, it means tissues are going to be lost. And if tissues are lost, it means there is no interconnection between one neuron and another, and therefore this can lead to memory loss. So death of cells or destruction of cells due to the breakdown of their membranes leads to the loss of tissues, and this loss of tissues leads to failure for different neurons to connect. That can lead to memory loss. Last part, which is question F, says, the team found genetic material from P. gingivalis in the cerebral cortex, which is paragraph 13. They say describe how the team could show that this genetic material is active. So again, let me read paragraph 13. It says the team also found genetic material from P. gingivalis in the cerebral cortex, a region involved in conceptual thinking. In all three Alzheimer's brains, they looked for in it. They look for it in. So what you have to do is, if you're looking for areas that are active in the brain or in any material, here we have to extract. We have to extract the infected part of the cerebral cortex, and then we will be able to get the cells and get out basically whatever is inside the nucleus. So we will use oligonucleotides compound with microarray, and then we can identify messenger RNAs present in the sample. Remember, any messenger RNAs present in the sample are indicative of any active genetic material, any active DNA or segment of the genes that are active and therefore are being transcribed. So if messenger RNA is present, it means there is a possibility that it's going to be used to make proteins. So those proteins could be the ones we are looking for. So we will analyze the messenger RNA in the sample and identify which genetic material these messenger RNAs are coming from. So, and then we can use bioinformatics to analyze the enormous quantity of data generated from the microarray and be able to connect and make a conclusion on whether this active material is due to the infection that has occurred or in the cerebral cortex or not. So this brings us to the end of question 8 as well as the end of this paper. Thank you so much for being with us. We look forward to always sending out papers for you so that we can help your academic journey to be made easier. Please do remember to subscribe to our channel and see you in our next video. Bye-bye.